Hello everyone. Uh, we have already started with this chemical sterilization, and the next method used in the chemical method of sterilization is liquid sterilization. In liquid sterilization, generally two different types of liquids are used. First one is alcohol and second one is phenol. Now, alcohols are not sterilizing agent but generally used as a disinfectant, and it is most effective disinfectant for many reasons, as it can vaporize quickly without leaving any residue. Now, while performing different types of practicals in the microbiology lab, we use alcohol to sterilize or to disinfect the surface, and it gets it gets quickly evaporated, so the area gets cleaned in the lesser time, and they are capable of dissolving in the different types of liquids, which makes it effective against the lipid wrapped viral cells such as HIV and hepatitis A. And different types of viral cells are there. To remove such type of viral cells, alcohol is very effective, and they are inexpensive and very easy to handle. Although their vapors are flammable, we can easily use these alcohol for the disinfection of any particular surfaces. Different forms of alcohol such as ethanol and isopropyl alcohol, both are the members of alcohol family, and they can also show the similar disinfectant properties. Generally, ethanol is present in various alcoholic beverages, and isopropyl alcohol is also known as isopropanol or 2-propanol or rubbing alcohol. When these both the types of ethanol or isopropyl alcohol generally used in the 70% concentration in water, they can be used or highly effective as a disinfectant agent. So these alcohol can be used for the cleaning or the removal of microorganism from any particular surfaces. Second one is phenol. Now, phenol is generally used as an antiseptic agent. Various surgical equipments are there, for example scissors, knives or syringes, which are used in while performing different surgeries. Such surgical equipments are disinfected or sterilized by using phenols, and it is a oldest antiseptic agent. Now, phenol acts. How does phenol act? It damages the cell membrane and thus releasing the cell content and causing lysis. And what does this phenol do? It damages the cell membrane of bacteria or microorganism. When the cell membrane of this microorganism is damaged, all the cell components comes out of the cell, and ultimately the cell gets damaged or lysis or breakdown of that cell is caused. This phenol is generally found in various materials, for example mouthwashes, scrub soaps and surface disinfectants. Now, generally various disinfectants or room cleaners are there, mouth washes are there, or different types of soaps and detergents are there. In all these materials, phenols are present, and they are used as a decontamination for the decontamination purposes. various hospitals or in laboratories for the cleaning purpose or disinfectant purpose, phenols are generally used. These were the liquids or the chemicals used for the sterilization purpose. Next method is your last method of sterilization, which is filtration method. Now, those material which are heat sensitive or sensitive to the steam or different types of chemicals, such sensitive materials are simply filtered through different types of filters for the sterilization purpose or removal of microorganism. Now, in filtration method does not kills the microorganism, but it simply removes or lowers the microbial count of any particular liquid substances. Now, this filtration, it is completely depend on the size of the organism. Now, this filtration process is very simple. The material whichever we have to sterilize is passed through the filters and it is collected in the completely sterilized receiver. If the receiver or any particular container in which we are collecting filtrate is not sterilized, then there are the chances of contamination. So the receiver should be completely sterilized. Now there are different modes of actions of filters. For example, sieving and screening, entrapment, electrostatic attraction. Means when a particle is larger than the pore size, मतलब हम जो भी filter process कर रहे हैं, if the particle which we want to filter is larger in size than the pore size of filter and that particle remains on the filter, then this process is called as sieving or screening. Means when a particle is larger than the pore size of the filter, the particle is retained on the filter. This is known as sieving or screening. Then the next one is entrapment. Entrapment means to hold or to stop any particular thing. इसमें क्या होता है, पोर साइज जो होती है वो पोर साइज लार्जर होती है पार्टिकल से and the channel is of lesser size than the pore size. मतलब यहाँ क्या होता है कि जो भी पार्टिकल की साइज होती है वो पोर साइज से छोटी होने के कारण वो पोर में एंटर तो करता है, लेकिन उसका जो चैनल है जहाँ से वो पार्टिकल पास होने वाला है वो चैनल की साइज छोटी छोटी हो जाती है, इसलिए वो जो पार्टिकल है वो पार्टिकल उस पोर में ही ट्रैप हो जाता है, वो उस होल में ही स्टॉप हो जाता है और फिल्ट्रेट जो है लिक्विड सब्सटेंस जो है वो रिसीवर में कलेक्ट किया जाता है. यहाँ हम क्या करते हैं, पार्टिकल को स्टॉप करते हैं या फिर ट्रैप करने का काम करते हैं. सो दिस प्रोसेस इज नोन एज एंट्रैपमेंट प्रोसेस. And the next mode of action is electrostatic attraction. Now the name itself indicates the particles are attracted and absorbed at the surface of filter bed, which is oppositely charged. मतलब अगर हमारा पार्टिकल पॉजिटिवली चार्ज होगा तो फिल्टर नेगेटिवली चार्ज यूज करके हम क्या कर सकते हैं, वो पार्टिकल को अट्रैक्ट करके फिल्टर को ही फिल्टर में ही उसको absorb करते हैं या फिर stop करते हैं, so that filtrate will move away and the particle will remain on the filter. These were the mode of action of any particular filter. मतलब हम sieving or screening method we can use. Then we can use entrapment process or electrostatic attraction. Now there are 4 different types of filters used for in the sterilization process, in which first one is your membrane filter. Now in your SY practicals you might have already used the membrane filter. Generally the membrane filters have the pore size less than 0.45 micrometer. And the bacterial size is more than 0.5 micrometer. So the liquids or the oily substances which contains the microbial count are filtered by using membrane filtration technique. यहाँ क्या किया जाता है? The assembly of membrane filter is set, and these oily or liquid substance is passed through those filter. The bacteria or the different microorganisms get trapped or stopped above the filter, and the pure sterilized liquid or the liquid which does not contain any microbial load is collected inside the receiver. But the organic substances like alcohols and chloroforms are generally not filtered by using membrane filtration technique. See now here in this diagram we can see the membrane filter. Here we can see that in the upper surface the oily substances or the liquid containing substances are generally passed, and here in the red area we can see that membrane filter is set, and downside it the sterilized receiver is available. When the liquid is passed through this filter, all the microbial load is trapped on the filter and a clear liquid is passed inside the receiver. Then next one is sintered or fritted glass filter. यहाँ क्या किया जाता है? Ball mill. Here we can see in the diagram, ball mill is seen. इस ball mill में क्या किया जाता है? The borosilicate glass is finely powdered. Borosilicate glass जो होता है उसकी fine powder की जाती है. And these fine powdered borosilicate glass of required size is generally separated and added in this ball mill, and it is heated till it gets completely fused. यहाँ क्या किया जाता है कि ये जो भी बॉल मिल है उस बॉल मिल में बोरोसिलिकेट ग्लास की जो पाउडर है वो पाउडर ऐड करके उसे हीट किया जाता है. इट इज हीटेड टिल इट गेट्स completely fused, and when it gets completely fused the pore size of that layer is generally 2 millimeter. Then after fusing it, the sulfuric acid, our filtrate is passed through these ball mill, and filtrate is collected in the receiver. फिर क्या किया जाता है? एक बार ये ग्लास फ्यूज्ड होने के बाद हमारा जो भी कंटेंट है लिक्विड कंटेंट हम इसे क्या करते हैं? फिल्टर कर लेते हैं. द पार्टिकल्स हैविंग पोर्स साइज more than 2 millimeter are trapped on this glass and remaining filtrate is passed through it. बाद में क्या किया जाता है? After this process is completed, this ball mill is cleaned with the help of sulfuric acid. Next one is your SIDS filter. SIDS filter you can see in this diagram. It generally contains 2 parts. The lower part is filled with the perforated plate, मतलब प्लेट जिसमें different types of pores present होते हैं, and it is compressed with the asbestos pad which is placed on it, and the upper part has a wall through which the pressure can be applied. मतलब नीचे के पार्ट में क्या होता है, एक परफोरेटेड प्लेट होती है और उस परफोरेटेड प्लेट के ऊपर अपर पार्ट सेट किया जाता है जिस पर एक वॉल होता है और उस वॉल से प्रेशर अप्लाई किया जाता है. Then these both the parts are joined together by using different types of hinged nuts or packaging material, and then our filter is passed through it, or the material is passed through it for the filtration process. The main advantage of using SITS filter is that there is low risk of contamination, and it is easier to use, and highly viscous solution can also be filtered by using SITS filter. Then next one is ceramic filter. Now ceramic filter is inexpensive and it is highly effective type of water filter. Generally ceramic filters are used for the filtration of water, and it has small pore size, so through this all the dirty materials or the debris or the bacterial count whichever is present inside the water can easily filtered, and water can be used for the various purposes. But the disadvantage of using ceramic filter is it is not effective against viruses, means as viruses have very small size, they can easily get filtered or can easily pass through the pore size of the ceramic filters, and they cannot be removed. And the different types of chemical contaminants can also not be removed by using ceramic filters. So this ceramic filter is only used for removing the dirty materials or the debris present inside the water. And various manufacturers, what they do, they add high performance activated carbon core inside the ceramic filter, which reduces the organic and metallic contaminants. मतलब organic और metabolic contaminants को remove करने के लिए this activated carbon को क्या करता है, ceramic filters में add किया जाता है, so that these contaminants can be easily removed. Now here are two types of ceramic water filter. First one is your pot type ceramic water filter, and second one is your candle type of filter or candle type of water filter. So this was all about the methods of sterilization or the different types of methods used for the sterilization process. We have already seen three main types of sterilization method. First one was physical method. In physical method we have seen the four subtypes of methods used for the sterilization, which was sunlight, then radiation, dry heat sterilization, in which again we have seen two types. Next one was your chemical methods used for the sterilization process, in which gaseous, we have seen gaseous sterilization and liquid sterilization. And the last method was your filtration technique, in which we have seen the four different types of filters used for the removal of microorganism and sterilization of your product. For this purpose you can use different types of references, or you can use Principles and Practice of Disinfection and Sterilization book by A.P. Fress. Thank you.